Imagine a future where kidney failure could be halted in its tracks for thousands of patients. That future might be closer than you think. A groundbreaking study presented at the 2025 American Society of Nephrology Kidney Week has revealed promising results for a new treatment targeting focal segmental glomerulosclerosis, FSGS, a condition that often leads to kidney failure within a decade for about half of those affected. But here's where it gets controversial. Could targeting a specific protein channel in the kidney cells be the key to slowing, or even stopping, this devastating progression? FSGS is a complex disease where the kidney's filtering units, called glomeruli, become scarred, leading to proteinuria, protein in the urine, and, eventually, kidney failure. Research has pinpointed the overactivity of the transient receptor potential cation channel, subformula C, member 6, TRPC6, as a major culprit. This channel's hyperactivity contributes to the loss of podocytes, specialized cells essential for kidney filtration, and accelerates the decline in kidney function. Interestingly, inherited mutations in TRPC6 that make it overly active are directly linked to genetic forms of FSGS, making it a prime target for therapy. During the conference, Dr. Nicholas Cross and his team unveiled the results of a Phase 2 clinical trial that tested BI-764-198, an oral medication designed to block TRPC6. The study, a multicenter, double-blind, placebo-controlled trial, aimed to determine whether this drug could reduce proteinuria in FSGS patients. And this is the part most people miss. The trial specifically focused on patients with both primary FSGS and genetic FSGS caused by TRPC6 mutations, offering a tailored approach to treatment. The primary goal was to see if BI-764-198 could reduce proteinuria by at least 25% compared to baseline levels after 12 weeks. Secondary objectives included assessing the drug's safety, tolerability, and how it behaves in the body. Patients were randomly assigned to receive either 20 mg, 40 mg, or 80 mg of the drug daily, or a placebo, while continuing their standard kidney-protective and immunosuppressive treatments. Out of 60 patients who completed the trial, 7 had TRPC6 mutations. The results were striking. At the 12-week mark, 44% of patients on the 20 mg dose, 14% on the 40 mg dose, and 43% on the 80 mg dose showed significant proteinuria reduction, compared to just 7% in the placebo group. Even more remarkable, all 4 patients with TRPC6 mutations who received BI-764-198 achieved a proteinuria response, while none in the placebo group did. The 20 mg dose emerged as the most effective, with a placebo-corrected mean reduction in proteinuria of nearly 40%. Importantly, the drug was well tolerated, with no significant differences in side effects across the treatment groups. Researchers hailed these findings as the first evidence that a podocyte-targeted therapy can effectively treat FSGS. But here's the thought-provoking question: if TRPC6 inhibition proves to be a game-changer for FSGS, could it also open doors for treating other kidney diseases driven by podocyte dysfunction? And what does this mean for the future of personalized medicine in nephrology? The study, funded by Boehringer Ingelheim, marks a significant step forward in kidney disease research. However, it also raises questions about accessibility and long-term outcomes. Will this treatment become widely available, and how will it impact patients' quality of life over time? What's your take? Do you think TRPC6 inhibition could revolutionize FSGS treatment, or are there still too many unknowns? Share your thoughts in the comments below.